Something is already happening inside the chest, not a thought, something underneath the thought. A pressure without a source, a kind of interior weather that arrived before you could have explained why. You may have felt this before and called it loneliness, but that word is almost too small for what the biology actually does. Welcome, quietly, to the Sleepy Biologist. This channel exists because some of you keep coming back, because something about understanding the body from the inside makes the night feel less strange. To the members who make this space possible, and to those who have left a kind word or pressed a like at 2 a.m. without knowing quite why, it matters. It genuinely does. This episode exists partly because of you. Now, let's go somewhere most explanations don't bother going. There is a structure in the brain, ancient, small, sitting near the brainstem, called the anterior cingulate cortex. It does not care about your name or your relationships or the specific person whose absence is making your chest feel dense tonight. What it cares about is signals, and the signals it monitors for physical pain and the signals it monitors for social disconnection run, in significant part, through the same tissue. This is not a metaphor the brain is using. This is the actual wiring. Naomi Eisenberger, a social neuroscientist at UCLA, spent years mapping what happens in the brain during social rejection, the specific sting of being left out, or let go, or unresponded to. When she put participants through a virtual ball-tossing game designed to simulate exclusion and watched their brains under fMRI, the anterior cingulate lit. The same region that activates when you stub your toe or pull a muscle or press on a bruise. The brain was treating a social event as an injury, not like an injury. As one, it is worth sitting with that for a moment, because it changes the question. Most of us, at some point, have been told, directly or indirectly, that emotional pain is less real than physical pain. Softer, more manageable, less biological, and yet here is a region of neural tissue, measurably active, doing the same job it does when the body has been damaged. No metaphor involved, just a circuit running. The insula is involved too. That folded strip of cortex buried between the frontal and temporal lobes, the part of the brain responsible for what researchers sometimes call interoception, the monitoring of your own internal body state. Temperature, hunger, heartbeat, the general sense of whether the body is okay. In studies of people who score high on loneliness measures, insular activity is altered. The region functions differently, less connected to the networks that process the external world, more isolated from the systems that usually help predict what will happen next in a social situation. The inside and outside stop communicating the way they should, and something in the body begins to feel, vaguely, persistently, like it doesn't quite know where it is. There's something that I find quietly strange about this, and I'm not sure the science has fully resolved it either. The opioid system, the same system that produces endorphins when you exercise, the same system that certain painkillers hijack, also responds to social bonding. Specifically, it responds to the kind of touch that happens between people who are comfortable together. Slow, gentle, lingering contact. The kind that doesn't really exist with strangers. Specialized nerve fibers in the skin, see tactile afferents they're called, respond preferentially to this kind of touch. Not firm or fast touch, the soft, unhurried kind. And when these fibers fire, they send signals deep into reward and emotion processing areas. The opioid tone of the brain shifts. When this contact is missing, not just today but as a sustained pattern, those same receptors begin to notice. The mu-opioid receptor, which quiets down in the presence of connection, stays active, alert, waiting. Pain and belonging, it turns out, are not opposite experiences managed by different systems. They are two states of the same system. One side reads, safe, connected, release. The other reads, absent, unanchored, hold. And the body, your body right now, has been reading one or the other of those lines for most of your life, shifting between them, sometimes in the same afternoon. The chest feeling, the weight in the throat, the strange heaviness in the hands that sometimes accompanies a particular kind of missing. These are not vague. They're not just emotional residue. The vagus nerve, a long, wandering nerve that runs from the brainstem down through the throat, heart, and gut, carries signals between the brain and the body's major organs in both directions. When the brain interprets a social situation as threatening or isolating, vagal tone shifts, heart rate variability changes, the gut receives different input. The muscles of the throat and the upper chest, which are partly governed by the same cranial nerves, involved in facial expression and social communication, can carry a kind of tension that has no obvious physical cause. There is an actual reason the throat tightens, an actual reason the chest feels occupied by something that isn't air. The biology is not approximating the feeling, it is producing it. Somewhere further back, much further back, evolution had something to say about all of this. Social species, and humans are among the most intensely social animals that have ever existed, did not survive alone. The presence of other members of the group meant protection, shared warmth, information about food and danger. Absence from the group was genuinely, concretely dangerous. The brain learned this, not in any individual lifetime, across hundreds of thousands of years of selection pressure. What emerged was a system that treats social disconnection as a threat state, not symbolically, physiologically. The hypothalamic pituitary adrenal axis, the body's central stress response system, responds to loneliness the same way it responds to predators. Cortisol rises, not sharply, not dramatically, slowly. A quiet, sustained elevation that alters inflammation, disrupts sleep architecture, changes immune function over time. You carry this ancient calibration inside you. It was not designed for the specific texture of modern loneliness, the kind that can happen in a full apartment, or on a phone, or in a long relationship that somehow stopped being present. It was designed for a world where being separated from your group meant you might not survive the night. The system does not update for context, it simply reads, alone or not alone. And when it reads alone, it begins its preparations. It's not entirely clear, and I think it is worth saying this honestly, where exactly the sensation stops being neural signal and starts being what we'd call feeling. There's a point somewhere in that cascade of anterior cingulate activity and vagal shift and opioid receptor state where something becomes experienced, where a pattern of firing becomes a presence in the chest. That threshold is one of the genuine open questions in neuroscience. The biology can be mapped up to a point, and then the map ends, and there's just this. Whatever you're feeling right now, or have felt or will feel again, the tissue that registers it is real, the chemistry is real, the evolutionary roots are deep and documented, and yet the thing itself, the actual quality of the ache, remains stubbornly, quietly irreducible. Something else is worth knowing before sleep takes this further. The pain of loneliness, the kind that activates the same regions as physical injury, is not a flaw in the system. It is the system working, a signal, aversive enough to motivate action, ancient enough to have been conserved across millions of years of social animal evolution. It hurts because it was designed to hurt, not cruelly, practically. The body needed a reason to seek others, and the other reason it chose was this: make the absence feel like something, which it does, which it has for a very long time. There are other structures involved, other pathways. A cluster of dopamine neurons sitting near the brainstem that begins, quietly, to shift its activity when isolation extends, not toward pain exactly, but toward something stranger, a craving that doesn't know what it wants until connection arrives and the craving stops. That part comes later. For now, just the ache, the one with no wound, the one the body learned to carry a long time before you were born. There is a region near the base of your brain, not large, not dramatic in its appearance under a microscope, that has been doing the same job for an extraordinarily long time. Not memory in the usual sense, not language, not the kind of knowing you can retrieve on purpose. Something older than that. It's called the dorsal Raphe nucleus, a small cluster of neurons tucked near the brainstem, closer to where your skull meets your spine than to anywhere you'd associate with thought or feeling. Neuroanatomists sometimes describe its position as tucked, almost modest, which I find quietly interesting, that something so small, so unremarkably located, turns out to be one of the places where loneliness actually lives, not metaphorically lives, physically, in tissue. The dorsal Raphe nucleus is best known for serotonin. It's one of the brain's main serotonin-producing centers, which is why it shows up in discussions of depression and mood regulation. But tucked within it is also a distinct population of dopamine neurons. And these particular neurons, it turns out, have a very specific sensitivity. They notice when you're alone. The key study came out of MIT in 2016, published in Cell, which in neuroscience terms means the finding was considered significant enough to withstand a great deal of scrutiny. Researchers were working with mice, using a technique called optogenetics, the ability to switch specific neurons on and off using pulses of light delivered through a tiny fiber optic cable threaded into living brain tissue. It's a strange image, honestly. A mouse moving freely around a cage with a thin thread of light reaching into its brainstem. But the technique allows a precision that nothing else currently offers. You can ask what happens when exactly these neurons fire, and you can ask the opposite. What happens when they don't? What the researchers found was this. When mice were socially isolated, even briefly just overnight, the dopamine neurons in the dorsal Raphe nucleus underwent synaptic strengthening. The connections feeding into them became more excitable, more ready. And when those mice were subsequently introduced to a new mouse, the DRN dopamine neurons fired strongly upon social contact. The animals sought out interaction with an urgency that non-isolated mice simply didn't show. The isolation had primed something, made it hungrier. And here is where the hunger metaphor becomes almost uncomfortably literal. A follow-up study, this one in humans, led by Rebecca Sachs's group at MIT, asked participants to do two things on separate days: fast for 10 hours, and then, on another day, be completely socially isolated for 10 hours. No phone, no contact, no interactions of any kind. After each depression, participants were placed in a brain scanner and shown images of food, images of people socializing, and neutral images, flowers used as a control. The region that responded to images of socializing after isolation was the substantia nigra, a small dopaminergic structure in the midbrain that shares deep evolutionary roots with the dorsal Raphe nucleus. The same region lit up for food images after fasting, not similar regions, not neighboring regions, the same one. The brain was using identical neural real estate to represent two entirely different kinds of wanting. I keep returning to that, actually. The fact that the craving for company and the craving for food aren't just like each other. They are at some level running on the same substrate. There's something almost disorienting about it. We treat hunger as elemental, biological, undeniable. We would never tell someone that being hungry is a personal failing or a sign of weakness, and yet the same tissue, the same dopaminergic machinery, generates the want for another person's presence. Maybe the distinction we draw between physical needs and social ones is less solid than we've been led to believe. Or maybe it's just that the brain, across millions of years of building social animals, found an efficient solution. It already had a system for tracking deprivation and generating motivation. Why build a new one? The dorsal raphe of Nucleus doesn't only talk to one place. Its dopamine neurons project outward, to the nucleus accumbens, to the prefrontal cortex, to the amygdala, to structures involved in memory and threat detection, and the regulation of mood. When those neurons shift after isolation, the effects aren't contained. They ripple. This is part of why loneliness doesn't feel like it's coming from one place. It isn't. It spreads outward from a central signal through networks that touch nearly every domain of how you perceive and respond to the world. Your threat sensitivity shifts. Your interpretation of ambiguous social signals shifts. Your motivation shifts. Your sleep shifts. And yet the origin, or one of the origins, anyway, is this small cluster of cells near the brainstem that noticed you were alone and started preparing for reunion. There's something worth pausing on in that framing. The DRN dopamine neurons don't activate during isolation itself. They activate at the moment of social contact after isolation. The longing builds quietly, invisibly during the period of aloneness, and then when connection arrives, the neurons fire. Strongly, almost urgently. The researchers describe this as analogous to the rebound eating that follows a fast. The hunger accumulates in the absence, but the consumption, the relief, happens at contact, which means that the brain during loneliness is not simply registering pain, it is accumulating want. A side observation, and I'm genuinely unsure what to make of this. In the optogenetic experiments, when the DRN dopamine neurons were artificially activated, switched on by light, the mice showed two seemingly contradictory behaviors simultaneously. They sought out social interaction more, and they also showed avoidance behavior, place aversion, as if the state being induced was both a pull toward others and an underlying discomfort, both at once. Loneliness as a push and a pull, encoded in the same neurons. If that maps onto anything in human experience, I think most people who've felt genuinely lonely for a long time will recognize it. The wanting to be with people, and the strange resistance, the bracing quality, that makes it hard to actually close the distance. The DRN doesn't resolve that contradiction. It seems in some way to be the source of it. Something the chapter hasn't mentioned yet and probably should: social rank. In the MIT study, the degree to which DRN dopamine neurons modulated behavior was predicted by the animal's social rank. Higher-ranking animals showed stronger DRN responses to social contact after isolation. The neurons were more sensitive, more responsive, in individuals who had more to lose socially, or more to gain from reunion. This is still being worked out in human research, but it suggests that the loneliness signal isn't uniform across people. It's calibrated, at least partly, by social history, by what the nervous system has learned to expect, to want, to miss. The DRN is not just responding to the present, it's responding to something accumulated, a kind of cellular record of what connection has felt like and how much the body has come to depend on it. That word, remember, is doing something unusual in this context. The dorsal raphe nucleus doesn't remember the way the hippocampus does. It doesn't store episodes or facts, but the synaptic strengthening that follows isolation is itself a form of encoding. The neurons become more responsive because of what they've recently experienced. They carry the trace of absence in their changed connections. Not memory as narrative, memory as altered sensitivity. The brain doesn't forget that you are alone. It adjusts. And those adjustments, in how readily the DRN fires, in how strongly it projects to the structures downstream, persist beyond the isolation itself. The body takes time to recalibrate. The cellular record of loneliness doesn't erase the moment someone walks into the room. Somewhere in the midbrain right now, a population of neurons is in some particular state of readiness or quiet, shaped partly by tonight, partly by last week, partly by patterns that go back further than that. They're not doing anything dramatic. They're just calibrated, waiting in the particular way that living tissue waits, not patiently exactly, but persistently. And in the brainstem just above, the dorsal raphe hums with its ancient modest purpose, making serotonin, monitoring aloneness, keeping its own kind of time. The substantia nigra is not a region most people have heard of. It sits in the midbrain, small, dark actually. Its name means black substance in Latin, which refers to the neuromelanin pigment that gives it a visible colour in postmortem tissue. Most mentions of it in popular science involve Parkinson's disease because it's the region whose dopamine neurons are gradually lost in that condition. What it doesn't usually get credit for is this. It also tracks what you're missing. The MIT study, Tomova and colleagues 2020, published in Nature Neuroscience, is one of those papers that lands quietly and then keeps mattering. Participants were brought in on 2 separate days. On one they fasted for 10 hours, no food, nothing caloric. On the other, they were socially isolated for 10 hours, no phone, no conversation, no eye contact. The researchers were careful about this. It wasn't mild isolation, it was a genuine attempt to remove social input entirely. Then brain scanning. Participants were shown images, food, people socializing together, flowers, neutral, there to give the brain something unremarkable to process. And what the scan showed was that the substantia nigra responded to images of social connection after isolation the same way it responded to images of food after fasting. Not approximately, not in a loosely similar pattern, the same region, the same signal type, the same structure lighting up for 2 different absences. There's something about that finding that I've never quite gotten used to, even reading it multiple times. We accept hunger as involuntary and no one chooses to feel it. No one is blamed for it. No one is expected to simply decide not to need food. It is understood as a biological fact. And yet the same neural substrate running the same basic operation generates the felt pull toward other people. The brain doesn't have a separate lesser system for social wanting. It recruited the same machinery. Which means, and this is the part the study doesn't say outright but seems to imply, that the experience of missing someone, the specific interior pressure of it, may be as involuntary as being hungry, as biological, as outside the realm of personal failing, outside somewhere a refrigerator hums or a radiator ticks. The particular quiet of late at night has its own texture, separate from silence. The dopaminergic system involved here is an old one. Dopamine itself is not a uniquely human molecule. It appears across vertebrates, across invertebrates in systems that predate the development of social behavior by a very wide margin. What evolution did over time was repurpose. The same chemical signaling that originally tracked food availability and motivated foraging got recruited gradually into tracking social availability and motivating connection seeking. This is not a clean or deliberate process. Evolution doesn't design, it accumulates. And what accumulated in social species was a nervous system that treats the absence of other bodies with something structurally similar to the response to the absence of food. Not identical. The downstream effects differ. The timescales differ. The textures of the two experiences are nothing alike when you're actually inside them. But at the level of the substantia nigra, at the level of dopaminergic signal and what triggers it, the resemblance is close enough that a brain scanner, looking only at that region, might not immediately know which deprivation it was measuring. What I find stranger still, and this is maybe an odd thing to notice, is the flowers, the neutral control images, the flowers that failed to activate the substantia nigra regardless of which deprivation the participant was in. Not after fasting, not after isolation, the brain was unmoved. Flowers were flowers, but food was food, and people were people, and after 10 hours of being without either, the same region woke up for both. There's something clarifying about that null result. It suggests the response isn't general craving, isn't some diffuse state of deprivation-induced wanting. It's specific. The substantia nigra recognized the relevant category and responded to it. Social images read as social. Food images read as food. And the mechanism underneath, the dopaminergic tissue, treated both with the same basic urgency. The word craving is worth staying with for a moment because it usually implies something that can be resisted. Willpower, discipline, the choice not to act on a want. But the craving being described here is sub-behavioral. It's happening at the level of midbrain dopamine neurons before any decision is made. Before any awareness, possibly. The signal precedes the feeling, precedes the choice. And what generates the signal is simply time spent without the thing the system has learned to need. In that sense, it isn't craving in the colloquial sense, it's closer to a reading, a measurement the brain is taking. How long has it been? How much has accumulated? And then, without waiting for an answer from you, beginning to prepare. There are differences, of course, between food hunger and this. Food hunger has a relatively clear resolution. Eat, and the hypothalamic signals shift, the stomach sends satiation cues, appetite reduces. Social hunger is less clean. A conversation can satisfy it partially, a text can take the edge off briefly. The presence of someone you don't particularly feel connected to might do almost nothing. The system is sensitive not just to the presence of people, but to the quality of contact, or at least that seems to be the case, though the neural specifics of how the brain distinguishes meaningful connection from its absence are still being worked out, which is genuinely unsatisfying as an answer, but it seems honest. What the study gestures toward, and what subsequent work on the dorsal raphe and substantia nigra has continued to suggest, is that the social drive is not a soft preference layered on top of more important biological needs. It is structured like a biological need, tracked by ancient tissue, signaled through dopamine, motivated by deprivation, satiated imperfectly, variably by contact. Your body does not experience the absence of others as an inconvenience or a mood. It experiences it the way it experiences running low on something it requires. Not dramatically usually, not with alarms, just a quiet, persistent pull in a direction you already know. The substantia nigra keeps its own account, patient and dark and chemically precise, tallying something it has no name for, only a signal, only a state, only the particular readiness that builds when what the body expects is not there. And somewhere adjacent to that, other structures are beginning to adjust. The threat systems are waking up in their slow way. The stress axis is recalibrating. The brain is preparing without asking permission for a world it has decided might be less safe than it was before. There is a moment, you probably know it, even if you've never thought to name it, when someone you trust puts a hand on your shoulder, or pulls you close, or simply sits near enough that you can feel the warmth of another body. And something lets go, not a decision, not a conscious release, something underneath the decision-making, underneath the language, just loosens. A tension you may not have noticed you are holding. That loosening has a molecular address. The endogenous opioid system is one of the brain's oldest pain management structures. Endogenous means the body makes it. These are not external chemicals, but molecules produced internally, binding to receptors distributed across the brain and spinal cord and gut. The mu-opioid receptor is the primary one. It's the same receptor that morphine binds to, the same one that synthetic opioids target. When endorphins, the body's own opioid molecules, and attached to it pain quiets, distress softens. What is less commonly discussed is what this system does in the context of social bonding. In the presence of close physical contact, specifically the slow, gentle kind, endogenous opioids are released. The mu-opioid receptor activation shifts, and the effect isn't only pain reduction. There is a broader softening of threat response, a change in how the nervous system is reading the immediate environment. Safe, known, near. The opioid system, in this context, is not reacting to injury. It's reacting to its absence. It quiets because nothing is wrong. Robin Dunbar, an evolutionary psychologist at Oxford, has spent considerable time studying the biology of social bonding and argues that the endogenous opioid system is one of the central mechanisms through which social bonds are maintained in primates. Grooming, the physical contact that occupies enormous amounts of time in primate groups, produces measurable opioid release. It is, in a very literal sense, one of the ways the social brain rewards closeness. Humans do something analogous, not grooming exactly, but touch, casual contact, the kind that happens between people who have stopped being careful around each other. Here's the part that interests me, and that I think gets underemphasized in most discussions of loneliness. The mu-opioid receptor doesn't go dormant when contact is absent. It remains active, waiting. And in that waiting state, in the absence of the endogenous opioids that social contact would normally trigger, the system maintains a kind of baseline sensitivity that reads, in the body's internal accounting, as something adjacent to low-level pain. Not sharp, not acute, nothing that announces itself, just a persistent muffled signal that something is not being resolved. A 2003 study by Naomi Eisenberger and Matthew Lieberman, the same researchers who mapped social rejection onto the anterior cingulate cortex, found that opioid antagonists, drugs that block mu-opioid receptors, increased feelings of social disconnection and reduced the emotional warmth people reported feeling in social interactions. They weren't in pain, they weren't isolated. They were simply in a room with other people, with their opioid receptors blocked. And something was missing, chemically missing. The warmth that normally comes from social presence, that particular quality of ease, depends, at least in part, on this system working. Which also means when the system isn't being regularly supplied with the social contact that triggers it, the warmth doesn't fully arrive. People can be in the same room, having the same conversations, and something still feels faintly absent. Not because connection isn't happening, but because the receptor is waiting for a signal that requires more than proximity to activate. It requires something more like safety, familiarity, the specific quality of contact that the C-tactile afferents in the skin evolved to detect. The C-tactile afferents deserve a moment here, because they are strange and specific and not widely known. These are unmyelinated nerve fibers found in hairy skin. Arms, back, the sides of the face. They respond preferentially to light, slow stroking touch at a velocity of roughly 1 to 10 centimeters per second. Not pressure, not fast contact, the particular pace of a hand moving gently. When stimulated, they send signals along pathways that bypass the usual somatosensory cortex, the part of the brain that processes ordinary touch, and project instead toward the posterior insular and limbic structures. Regions involved in emotional processing. Social reward. They are in effect a dedicated social touch system, built not to detect texture or temperature or pain, but to register the specific signature of deliberate caring contact. And when that contact doesn't come, sustained over time, these fibers have nothing to report. The insula receives no input from this channel. The limbic system waits. Something quiet in the body keeps waiting. The kappa opioid receptor tells a different story. Where the mu opioid receptor is involved in comfort and social reward, the kappa receptor is associated with dysphoria, a persistent low-grade state of unease. Social isolation in animal models activates kappa opioid signaling. The molecule that binds preferentially to it, dynorphin, is released under conditions of stress, pain, and, relevant here, prolonged social deprivation. So the opioid system under loneliness isn't simply quiet, waiting for input. Part of it is actively generating a signal, not loud, not acute, but present. The mu receptor underlights the warmth that should be there. The kappa receptor produces something to fill the space. 2 receptors, same molecular family, operating in opposite directions and both of them shaped by whether other people are close. I find something almost unbearably precise about this. Not sad exactly, just precise. The body built a system that rewards social contact with its own internal chemistry, and built a corresponding signal for its absence. It didn't leave a neutral state in between. There is no opioid equivalent of indifference to connection. You are either receiving the signal, or you're noticing its lack. The loosening when it comes, the shoulder, the hand, the warmth of someone close, is this system resolving. Mu opioid receptors binding, kappa signaling quieting, the insula receiving finally what the C-tactile fibers have been calibrated to send. It happens in seconds. The chemistry doesn't wait for you to process the moment emotionally. It responds before you do. Which is part of why touch, genuine and unhurried, can shift something faster than words. Not because it means more, it doesn't necessarily, but because it speaks directly to a system that predates language by a significant evolutionary stretch and has never learned to respond to sentences. Right now, in whatever position your body has settled into, your skin is in contact with something. Fabric, a surface, your own weight. The C-tactile afferents are reporting or not reporting depending on the nature of that contact. The mu opioid receptors are in whatever state they're in. The kappa signal is at whatever level it's at tonight. You don't have access to these numbers, but your body is running them quietly as it always does, maintaining its account of what it has received and what it still, in its cellular and ancient and completely unsentimental way, needs. The hypothalamus is not large. In a human brain it weighs roughly 4 grams, sits just below the thalamus, just above the brainstem, about the size of an almond, though that comparison gets made too often, and I'll only use it once. What matters is not its size but what it monitors. Hunger, thirst, temperature, circadian timing, reproductive state, and relevant here, threat. Not dramatic threat. Not the acute kind that makes you jump. The slow kind. The ambient kind. The kind that accumulates when the social environment stops feeling reliable. When the hypothalamus detects threat, any threat, physical or social, acute or perceived, it releases a small peptide called corticotropin-releasing hormone, or CRH. This travels a very short distance to the pituitary gland directly below. The pituitary responds by releasing adrenocorticotropic hormone, ACTH, into the bloodstream. ACTH travels to the adrenal glands sitting above the kidneys and there triggers the release of cortisol. This is the HPA axis. Hypothalamic pituitary adrenal. It is one of the most studied pathways in all of stress biology, and it does not care what kind of threat initiated it. Lonely individuals across multiple studies show dysregulated cortisol patterns, not always elevated in the morning, not always spiked at any particular measurement point. The disruption is more subtle than that, a flattening of the diurnal curve, meaning the normal steep decline of cortisol across the day becomes shallower. The system stays activated longer than it should. It doesn't fully power down, and at night, this is the part that compounds everything, cortisol remains higher than in non-lonely individuals through the sleep hours. The normal circadian drop, which the body needs to enter and maintain deep slow-wave sleep, is blunted. The body wanted to rest. The chemistry wouldn't fully allow it. There is something almost grimly efficient about the design, from an evolutionary standpoint. If you are separated from your group, genuinely separated, in the environment where this system was built, then sleeping deeply is dangerous. Reduced vigilance when you are exposed and alone is not adaptive. The HPA axis, which cannot tell the difference between ancestral isolation and a modern apartment, treats the social signal the same way regardless. Stay lighter, stay readier. Don't go all the way down. It isn't insomnia, it isn't anxiety in the clinical sense. It's the body doing exactly what it was designed to do in a context where that design no longer fits the actual danger level. Somewhere a door closes in another part of the building, muffled, distant, the kind of sound you only hear when everything else has gone quiet. Outside the HPA axis, something else is running in parallel. The sympathetic nervous system, the branch of the autonomic nervous system associated with arousal and threat response, shows what researchers call selective tonus in lonely individuals. Sustained low-grade activation, not the full fight-or-flight surge. Not anything that would register as distress if you checked in with yourself right now, just a slightly elevated baseline. A quiet readiness that the body maintains without asking. Heart rate variability, a measure of how flexibly the heart responds to changing demands, and a reliable indicator of autonomic nervous system health, is reduced in chronically lonely people. The heart beats with less variability, less responsiveness. The system is holding a note rather than moving through a range. John Cacioppo's lab found this in studies tracking lonely college students. Their cardiovascular reactivity was blunted, their stress responses were dysregulated, and crucially, the restorative behaviors they engaged in, sleep, exercise, spending time with others, didn't produce the same recovery effects they produced in socially embedded individuals. The biology wasn't absorbing the repair the way it should. This is the part I find most difficult to sit with. Not the cortisol, not the sleep disruption, though. Those are uncomfortable findings, but they feel comprehensible. What's harder is the suggestion that the repair mechanisms themselves become less effective. that loneliness doesn't just create a wound, it changes the healing environment. The same actions that restore a non-lonely person, a good night's sleep, a walk, even a social interaction, land differently when the underlying state is one of chronic social threat perception. The system is so thoroughly oriented toward alert that even recovery inputs get processed through that filter. Interleukin-6, an inflammatory cytokine, a signaling molecule released by immune cells, is elevated in lonely individuals independent of objective social isolation. This is important. It is not merely the absence of people that produces the elevation, it is the perceived absence. The subjective sense of disconnection. The immune system is reading a psychological state. IL-6 is not inherently harmful, it plays important roles in immune response and tissue repair, but chronically elevated levels are associated with cardiovascular disease, type 2 diabetes, accelerated cognitive decline. It is a molecule built for acute deployment, not sustained presence. And loneliness, it turns out, is one of the things that keeps it circulating. The inflammatory signal wasn't designed for this particular use. It was designed for infections, for wounds. The body borrowed the mechanism and applied it to something else entirely. There is a feedback quality to all of this that is worth making explicit, because I think it changes how the whole picture looks. Elevated cortisol disrupts sleep. Disrupted sleep increases threat sensitivity. Increases threat sensitivity amplifies the perceived social danger. Amplified perceived danger activates the HPA axis further. The HPA axis releases more cortisol. The loop doesn't require a catastrophe to sustain itself. It runs on mild, chronic biological unease, the kind that accumulates so gradually that no single day feels like the one where things changed. And the sympathetic nervous system is there throughout, not surging, not alarming, just maintaining its quiet, exhausting readiness, like a building where the emergency lights never quite go off, running on a separate circuit, dimly illuminating everything, costing something no one is accounting for. The word mild in the chapter title is doing real work. A body on mild alert is not a body in crisis. It functions. It gets through days. It goes to sleep and wakes up and moves through the world in ways that are largely indistinguishable from a body that is not on alert. The difference is internal and cumulative. The cortisol curve that never fully descends, the heart rate variability that stays compressed, the inflammatory markers running slightly hot, the sleep architecture that keeps pulling toward lighter stages. None of these is a diagnosis. All of them are, in aggregate, a biological cost. Paid quietly, paid continuously, paid in the particular currency of a nervous system that believes, at a level well below conscious thought, that the environment is not entirely safe. It doesn't announce this belief. It doesn't need to. It just holds lightly, persistently, in the particular way that living tissue holds things it has been given no clear signal to release. And somewhere in the adrenal cortex, cortisol waits for the instruction to quiet down. The instruction that, in a body keeping this particular vigil, may arrive later than the body needs it to, or may not tonight arrive at all. 120 milliseconds. That's not long. A blink takes roughly 150. So the thing this chapter is about happens faster than that. It's over before your eye has finished closing. Stephanie Cacioppo and her colleagues at the University of Chicago published a study using high-density electroencephalography, a technique that measures electrical activity across the scalp with enough precision to track the timing of neural responses down to the millisecond. They wanted to know not just whether lonely people processed social threats differently, but when. The answer was 120 milliseconds after the image appeared. That's when the lonely brain had already differentiated a social threat from a non-social one, had already begun routing it differently, already started responding. Non-lonely participants, around 250 milliseconds, more than twice as slow. The stimuli were images, social threats, exclusion, rejection, conflict between people, non-social threats, a snake, a nymph, something sharp and dangerous but not interpersonal, and neutral images used as a baseline. What the EEG recorded was the timing of neural differentiation. The moment the brain began treating the social threat image as categorically distinct from everything else. In lonely participants, that moment arrived early, very early. Before conscious awareness, before any decision about how to respond, before almost certainly any felt sense of having noticed anything at all. The technical term is implicit processing, meaning it happens below the threshold of intention. You are not choosing to scan the room for signs of rejection. The scan is already running, has been running, will keep running whether you want it to or not. There is a particular quality to this finding that I keep returning to. It isn't that lonely people are more fearful or more anxious or more damaged in some general way. The response is specific. It's tuned. Social threat, not all threat, not snakes, not sharp objects, but the particular signature of interpersonal danger is what gets fast-tracked. The brain has learned, through some combination of experience and biology, exactly what to watch for, and it watches for it first. The EEG data showed something else. In lonely participants, early recruitment of brain areas involved in attention and self-representation accompanied the fast threat detection. The self was implicated immediately, not just there is a threat, but something closer to there is a threat, and it involves me. That distinction matters. A non-social threat, the snake, doesn't immediately recruit self-representation areas. It's dangerous, but it's not about you in the social sense. The social threat is different. It carries, encoded in those early milliseconds, a kind of personal relevance that the nervous system has already decided is worth prioritizing. Outside, the night has that particular stillness it gets around 3 a.m, not silent, just settled. John Cacioppo's evolutionary model predicted this. The idea was that feeling socially isolated, being on the social perimeter, as he put it, should produce heightened surveillance of the social environment, because in the ancestral context, being on the perimeter was genuinely dangerous. You needed to know faster who was hostile. You needed to update your threat assessment before they reached you. 120 milliseconds is the nervous system doing exactly that. It's a very old adaptation running in a very new context. And the context it's running in now, a face glimpsed across a room, a tone of voice in a message, the ambiguous expression of someone you're not sure you can trust, doesn't require that speed. There's no predator. There's no group about to close ranks against you. But the timing doesn't adjust for context. It just fires. What this produces, practically, is a perceptual environment that is subtly but consistently skewed. Ambiguous expressions read as cold. Neutral tones carry an edge that probably isn't there. A delayed response to a message becomes evidence of something. The gap between what is actually present in a social situation and what the threat detection system is reporting can become over time significant, not imagined, not irrational, biological, running at 120 milliseconds upstream of reason. Quantum magazine's coverage of Danilo Bezdok's work at McGill described it as a downward spiral. lonely individuals receiving information through a system bias toward negative interpretation, which deepens the sense of isolation, which maintains the threat detection bias, which ensures the next social encounter is read through the same filter. The speed of the detection is part of what makes the loop hard to interrupt. By the time you're consciously processing the social moment, the nervous system has already filed its report. 120 milliseconds, less than a blink. The gap between what the world is and what a lonely nervous system has already decided it might be. Not a flaw, not a malfunction. A calibration, ancient, specific, and in this particular context, running just slightly ahead of everything else. There is a particular quality to feeling alone in a body. Not alone in a room, that's different, that's total. This is something more interior, a sense that the boundary between you and everything outside has become slightly less permeable. That the world is happening at a small but consistent remove. You can see it, respond to it, move through it. But something in the translation feels off, like sound through a wall rather than through open air. That quality has a neurological address, partial, incomplete, still being mapped, but an address nonetheless. The insular cortex, the insula, is a fold of cerebral cortex hidden from view, tucked behind the frontal and temporal lobes in a region called the lateral sulcus. You can't see it from the brain's outer surface, it sits inside a groove folded inward, which feels somehow appropriate for a structure whose primary job is monitoring the interior. Interoception is the technical name for what the insula does, the continuous, mostly unconscious tracking of the body's internal state. Temperature, heartbeat, the dull pressure of a full stomach, the specific quality of a breath that doesn't quite feel complete. The insula receives signals from the body's organs and viscera, integrates them, and generates what researchers sometimes call the sense of the physiological self, the felt sense of being a body that is currently alive and in a particular condition. This is not a glamorous function. It doesn't get written about the way memory or language does, but remove it or disrupt it, and something fundamental about the sense of being present in a physical self begins to fray. In lonely individuals, insular function is altered. The specific finding from neuroimaging research is a reduction in functional connectivity between the insula and the nodes of the attention network, the systems the brain uses to orient toward and track what matters in the environment. What this means, translated, the insula is still running, still receiving body signals, but its communication with the networks that direct attention outward is degraded. The inside and outside of experience, what the body is feeling and what the world is presenting, are less tightly coupled. Predictions become harder to make. When the insula feeds into the attention network, it helps calibrate expectations about the social environment. It allows the brain to anticipate in a low-level way what is likely to happen next with another person. When that connectivity is reduced, the calibration slips. The social world becomes slightly less legible. Somewhere in the building, water moves through a pipe, a sound with no source you could name, brief and unremarkable, gone before you fully register it. There is something I find genuinely strange about this, and I don't think the strangeness fully resolves. The insula is involved in empathy, in feeling pain on behalf of others, in the particular neural response that occurs when you watch someone else get hurt and something in you responds as if the hurt were yours. Studies of empathic response consistently show insular activation, the body monitoring system briefly treating another person's state as information about its own interior. So a structure that is disrupted by loneliness is also centrally one of the structures involved in feeling connected to other people's experience. The disruption doesn't eliminate this capacity. That would be too clean, too mechanical, but it does suggest that chronic loneliness might gradually shift how available that empathic response is, not as a character flaw, not as a choice, but as a downstream consequence of already insular connectivity. I'm not certain that's what the data shows. It might be, is the kind of finding that keeps accumulating support without ever becoming fully settled. The insular also does something that rarely gets mentioned in this context. It plays a role in the sense of time, specifically the subjective sense of duration. How long a moment feels, whether time seems to be moving or standing still. There is evidence, not vast but consistent, that insular activity contributes to the felt passage of time, to the difference between a minute that stretches and a minute that vanishes. Lonely people often describe time differently. Not always, not universally, but there is something in the phenomenology of loneliness that involves duration becoming strange. Evenings that go on too long, the specific texture of a Sunday afternoon that won't end. Whether this connects to insular disruption in any direct way, I genuinely don't know. The research hasn't traced that particular thread all the way through. But the insula sits at exactly the intersection of bodily monitoring, temporal processing, and social connectivity that would make it a plausible part of that experience. It is one of those places where the biology and the felt experience seem to be pointing at each other without quite touching. What the insula at its best does in social situations is something like this. It reads the body. It receives the signal from a racing heart, from a shift in breathing, from the specific muscular tension of being near someone unfamiliar. It integrates those signals and passes a kind of summary to the prefrontal cortex and the attention networks. This is what the body is currently registering in this situation. And that summary influences quietly, continuously, how the social moment is being interpreted. Is this person safe? Is this situation familiar? Does the body's response match what previous experience predicts? When the insula is well connected, this process runs smoothly. The body's response and the social interpretation stay loosely synchronized. When the connectivity is reduced, the synchrony degrades. The body might be registering something, unease, or the particular flatness that comes with prolonged isolation, and that signal is less efficiently reaching the systems that would incorporate it into a coherent social read. So the social world continues to be navigated, but with one of its most important feedback loops running at reduced bandwidth. The shape of other people, the way they feel as presences, the sense of their interiority, the quality of contact, is not processed by a single region or a single pathway. But the insula is part of how the body is kept informed about what another person's presence is doing to it. And when that system is altered, when the inside stops communicating cleanly with the systems tracking the outside, something in the quality of human presence changes. Not disappears, not stops mattering, just becomes slightly harder to receive, like a signal that is still transmitting but is arriving at the other end quieter than it should be. You already know this feeling, possibly. The way a room full of people can feel strangely muffled. The way a conversation can be technically present and yet somehow not land. The way you can be touched and feel the touch and still notice something is not quite arriving. The insula isn't the only reason for that, but it is part of the reason. And it is apparently one of the regions that loneliness changes first. The ventral striatum sits deep in the forebrain below the cortex, curled around the lower end of the caudate nucleus. It includes a structure called the nucleus accumbens, which has the distinction of appearing in almost every discussion of reward, motivation, pleasure, and addiction, which tells you something about how central it is, and also about how broadly the word reward gets applied in neuroscience. What the ventral striatum does, roughly, is track the anticipated value of things. Not just pleasure in the immediate sense, but the prediction of whether something is worth pursuing. Dopamine signals arriving here from the midbrain, from the ventral tegmental area primarily, update those predictions continuously. The system learns what has been rewarding before and orients toward it. In social animals, other people are among the most reliably rewarding stimuli available. The ventral striatum responds to faces, to social cues, to the presence of someone familiar. This is not a metaphor for connection. It is the mechanism partially underlying it. So what happens to this system in loneliness? The honest answer is, it depends on which study you read, and the studies don't fully agree. A 2009 study from Cacioppo's group found that lonely individuals showed reduced activation in the ventral striatum when viewing pleasant social images. Two people laughing, close physical contact, the visual signatures of warmth. Reduced. As if the reward signal had dimmed. As if the brain had, at some level, stopped fully expecting social images to deliver what they seem to promise, which makes a certain intuitive sense. If social experience has been consistently unsatisfying or consistently absent, the prediction gets updated. The dopaminergic signal that says this matters, pursue this, becomes quieter, not gone, but quieter. Then a 2016 study by Inagaki and colleagues found something nearly opposite. In lonely individuals, ventral striatal activation to images of close others was amplified, not dimmed, heightened. Compatible, the authors suggested, with a state of social craving. The brain was not blunted to social reward. It was reaching for it, responding to the image of closeness with something that looked, in the scanner, like want. Both findings are real. Both were peer-reviewed, both replicated in their own contexts. They simply point in different directions. There is a smell that comes through a window left open at night. Something green and faintly damp, difficult to name. It arrives and is gone before you can decide what it was. I've spent some time with this contradiction and I am not sure it fully resolves. But there is one way of holding it that feels to me more honest than forcing a single narrative. The two findings may both be correct because they were describing different things, or different people, or different stages of the same process. Early loneliness, or situational loneliness. The kind that arrives after a move, a loss, a disruption, might produce heightened ventral striatal response, the craving version. The system is still expecting reward from social contact and is responding to its imagery with something like urgent recognition. Chronic loneliness, sustained, long-term, woven into the fabric of daily experience, might eventually produce the dampened version. The prediction has been updated so many times, the reward signal has been sent toward connection so many times without adequate return, that the system begins to discount it. Not consciously, not as a decision, just, quietly, cellularly, it stops bidding as high. A 2019 study by D'Agostino and colleagues found no association between loneliness and ventral striatal activation at all, neither heightened nor reduced, just absent, which might mean the effect is context-dependent, or age-dependent, or that the sample was different in some important way. This is the part of loneliness neuroscience that I think is worth being honest about. It is not yet a settled field. The brain imaging studies involve relatively small samples. The measures of loneliness vary across studies. The stimuli used, which social images, which emotional valence, which degree of closeness depicted, differ enough to make direct comparison difficult. What is consistent is this: the ventral striatum in lonely individuals is not neutral, it is doing something different. The precise nature of that difference may depend on factors the studies haven't fully controlled for. What this means for the felt experience of loneliness is something the scanners can gesture toward but not fully capture. The particular quality of seeing a stranger and feeling briefly something stir, and then feeling it recede. The way a face across a room can produce something that isn't quite hope and isn't quite recognition but lands somewhere between them. The way images of people close together can feel, depending on the night, either like something you want or something you've stopped believing in quite the same way. Both of those responses are the ventral striatum doing its work. Both are the dopaminergic prediction system running its calculations about what social contact is worth and how likely it is to arrive. The nucleus accumbens doesn't know you personally. It doesn't know your history or your specific situation or which particular absences have been the hardest ones. It knows frequency, it knows return, it knows what has been rewarded before and at what rate, and whether that rate is currently meeting expectation, and it adjusts, continuously, without asking. That adjustment, whether toward yearning or toward a kind of resigned quiet, is part of what loneliness feels like from the inside, even when you can't name what specifically has changed. Something is always being calculated, below the level of awareness, about the social world and what it's likely to offer. The ventral striatum is one of the places where that calculation runs, and what it concludes night after night, encounter after encounter, slowly becomes part of what you expect from other people, whether or not you meant to expect it. The brain is not finished when you are born. This is known, repeated often enough that it loses its strangeness. But it is worth recovering that strangeness for a moment. The fact that the organ you are using right now to process these words was still being substantially shaped well into your 20s and continues to be modified in smaller but real ways for the rest of your life. Neurons form new connections. Existing connections strengthen or weaken depending on use. Myelin sheaths thicken around heavily used pathways and thin around neglected ones. The physical structure of the brain is, at every scale, a record of what has been experienced. This is neuroplasticity, and it is usually described as a gift. It is also an exposure. Grey matter refers broadly to the parts of the brain densely populated with neuron cell bodies, the computational tissue, the processing regions. White matter refers to the myelinated axons connecting them, the wiring. Both can be measured with structural MRI. Both show differences in people who score high on measures of chronic loneliness. The prefrontal cortex, specifically the dorsolateral prefrontal cortex, shows reduced grey matter volume in lonely individuals across multiple studies. This is not a dramatic reduction. It is not visible to the naked eye on a scan. It shows up in group comparisons, in average data, in the kind of finding that requires careful statistics to detect. But it is consistent, and the prefrontal cortex is not an incidental region. It is where social cognition is largely coordinated. The interpretation of other people's intentions, the regulation of emotional responses in social contexts, the capacity to hold multiple social possibilities in mind simultaneously, to consider that a person's silence might mean tiredness rather than hostility, that an expression might be ambiguous rather than rejecting. These are not abstract cognitive functions. They are the biological substrate of navigating other people without continuously misreading them. And the tissue supporting them is, in chronic loneliness, measurably reduced. Whether that reduction precedes loneliness or follows it, whether people with slightly less prefrontal volume are more vulnerable to loneliness, or whether loneliness over time depletes the tissue, is not fully established. The honest answer is probably both, in different proportions in different people, across different timescales. What the cross-sectional data can't do is show you the direction of causation. It can only show you that the two things, chronic loneliness and altered prefrontal structure, tend to arrive together. Outside, the quality of darkness has shifted almost imperceptibly. Not lighter, just different. The way the middle of the night has its own texture, distinct from early night or near morning. White matter tells a related story. Diffusion tensor imaging, a technique that tracks the movement of water molecules along axonal pathways and can infer the integrity of white matter tracks, shows reduced connectivity between the insular cortex and attention networks in lonely individuals. This was mentioned in the previous chapter in functional terms. Here the finding is structural. The wiring itself, not just its activity, is different. Nakagawa and colleagues published findings in 2015 showing white matter structure differences associated with loneliness in young adults. Not elderly adults, not people with decades of isolation behind them, young adults. The implication being that structural effects are not purely the accumulation of a lifetime. They appear earlier than that. The brain begins adjusting its architecture to match its social experience well before middle age. The prefrontal cortex is roughly behind the forehead. If you place 2 fingers just above the bridge of your nose and press gently, not hard, just resting, you would be approximately above it. Several centimeters of skull and meninges and cerebrospinal fluid between your fingers and the tissue in question. A small physical distance for something so involved in the management of your entire social world. I find that proximity strange in a way I can't entirely explain, that the structure deciding whether someone's tone of voice is threatening or just tired is sitting right now a few centimeters behind your face, being shaped very slowly by what your life has contained. The amygdala shows structural differences too, though the findings here are more variable across studies. Some report increased volume, consistent with heightened threat processing. Others find no significant difference. The variability likely reflects the same problem as the functional imaging literature. Loneliness is not a single state, and the amygdala's response to it probably depends on duration, severity, and individual differences in threat sensitivity that no single study fully captures. What seems more robust is the hippocampal finding. The hippocampus, involved in memory consolidation and spatial navigation, but also in regulating the stress response through inhibitory projections to the HPA axis, shows accelerated age-related volume loss in lonely older adults. The structure that helps break cortisol output appears to wear faster in people who have been lonely for a long time. The stress system that won't quiet down gradually affects the structure that would quiet it. There is something in the accumulation of these findings that asks for a particular kind of patience to sit with. Not despair, that's the wrong register entirely, but a kind of seriousness. The brain is a physical object. It has mass, dimension, metabolic requirements. It changes in response to experience the same way muscle changes in response to load or disuse. And the experience of chronic loneliness, sustained, below the threshold of crisis, just the steady daily absence of sufficient connection, leaves physical traces in it, not wounds, not damage in any irreversible sense. The brain retains plasticity throughout life, and there is evidence that social reconnection can shift some of these structural markers, but the traces are real. Grey matter is not a metaphor for anything. It is actual tissue, neurons and glia and blood vessels, and the intricate local circuitry of a region that has been doing its particular job for as long as you've been alive. When its volume is reduced in a specific area, that area has less computational resource available. Not none, less. The weight of solitude in this chapter's title is not poetic, It is approximately 4 grams of tissue behind your forehead, carrying in its structure, in its connectivity, in the state of its white matter pathways. A partial record of how much time you have spent feeling genuinely close to other people, and how much time you have spent without that. Sleep is not a single thing. This is worth saying plainly because the word flattens what is actually a structured biological process with distinct phases, each serving different functions, each requiring different conditions to occur. When you fall asleep, when consciousness loosens and breathing slows and the muscles release their held tension, you're not simply switching off. You're beginning a descent through stages, each deeper than the last, each doing something the waking brain cannot do for itself. The deepest staging, slow-wave sleep, sometimes called N3, is where the most essential restoration happens. Growth hormone is released, immune function consolidates. The glymphatic system, a network of channels surrounding the brain's blood vessels, becomes significantly more active, flushing metabolic waste products that accumulate during waking hours. The brain, in slow-wave sleep, is cleaning itself, doing maintenance that requires the relative stillness of deep unconsciousness to accomplish. And this is the stage that loneliness quietly and persistently tends to cut short. John Cacioppo's lab published a study in the journal Sleep in 2011, tracking lonely and non-lonely individuals across multiple nights using polysomnography. The full suite of sleep measurements, including brain waves, eye movements, muscle activity, breathing. What they found was not that lonely people slept fewer hours. The duration was roughly comparable. What differed was the architecture. Lonely individuals experienced significantly more micro-awakenings. Brief arousals, fragments of lighter sleep or near wakefulness lasting less than 15 seconds each, scattered through the night without bringing the sleeper to full consciousness. The sleeper would not remember them by morning, but the body registered them. The descent into slow-wave sleep kept getting interrupted. Depth kept being exchanged for surface. The mechanism runs through cortisol. At night, in a healthy sleep architecture, cortisol follows a circadian decline, lowest in the early hours of sleep, beginning to rise again toward morning in preparation for waking. This decline is not incidental. It is part of what allows the brain to pass through the lighter stages and settle into the deep ones. Slow-wave sleep requires a neurochemical environment that high cortisol disrupts. The HPA axis, running slightly hot in lonely individuals, maintaining its evolutionary conviction that the social environment is not fully safe, keeps cortisol elevated through the hours when it should be falling. The body tries to go deep. The chemistry pulls it back. There is a specific sensation that belongs to this. You may know it without having named it. The feeling of being almost asleep. The weight of the body becoming unfamiliar. Thoughts losing their edges, images arriving without sequence. And then something. Not a sound exactly, not a thought exactly, and a slight tightening, a return, and not full waking. Just a small withdrawal from the threshold. The depth receding before you reached it. That is, in some cases, the micro-awakening. 15 seconds of the brain deciding below awareness that it should be slightly more alert, that the environment bears monitoring, and then back down, or trying to go back down. But the architecture has been interrupted, and the slow-wave stage requires a run-up, a gradual settling that the interruption resets, so the body tries again. The glymphatic system is worth dwelling on here, because its discovery is relatively recent. The term itself wasn't coined until 2013 by Mike and Nedergaard at the University of Rochester, and its implications for understanding the cost of disrupted sleep are still being worked out. During slow-wave sleep, the brain's interstitial space expands. The channels through which cerebrospinal fluid flows around and between neurons widen. Waste products, including beta-amyloid and tau proteins, both associated with Alzheimer's disease pathology, are cleared at significantly higher rates than during waking. The brain uses the stillness of deep sleep to do housekeeping that cannot be done when it is busy processing the world. When slow-wave sleep is repeatedly fragmented, when the body is pulled towards surface before the deep work can be completed, this clearance is reduced, not eliminated, reduced. Over one night, the difference is probably small. Over months, over years, the accumulation becomes its own biology. Memory consolidation happens during sleep too, primarily during slow-wave stages and REM. The hippocampus replays the day's experiences, transferring information to cortical storage, strengthening some connections and allowing others to weaken. Emotional memories are processed. The amygdala and prefrontal cortex engaged in a kind of overnight negotiation about what a thing meant and how much weight it should carry going forward. In fragmented sleep, this processing is incomplete. Emotional memories don't get the same overnight softening. The sharp edges stay sharper. The threat signal from yesterday doesn't fully reduce by morning. A person wakes still carrying more of the previous day than the brain had time to properly file away, which for someone whose nervous system is already biased toward detecting social threat, already running its 120 millisecond surveillance, the incomplete emotional processing of sleep just adds more unresolved material to the next day's already skewed red of the social world. The loop is not dramatic. That is what makes it so persistent. Loneliness elevates cortisol. Elevated cortisol fragments slow-wave sleep. Fragmented sleep leaves emotional threat responses less regulated. Less regulated threat responses make social situations feel more dangerous. More perceived social danger maintains loneliness. Maintained loneliness keeps cortisol elevated. Around and around, quietly, every night, nothing catastrophic in any single cycle. Just the gradual weight of a process that never fully completes, a body that never fully releases into the depth it needs, a brain that wakes each morning having done less of its overnight work than it required. There is something I find genuinely affecting about this, and I don't usually use that word. The idea that the body, lying completely still in the dark, completely alone, is trying to go deep, trying to clean itself, consolidate, repair, and being pulled back again and again by a stress signal that believes incorrectly that this particular night requires vigilance. The body is not failing. It is doing exactly what a socially isolated organism's body should do, by the logic of its own ancient design. It is just doing it at a cost that compounds in ways the design never accounted for. By morning, the surface will have been visited many times, the depth less so. And somewhere in the prefrontal cortex, in the glymphatic channels, in the emotional memory traces waiting to be processed, something that should have been resolved in the dark hours is still quietly waiting. The immune system is not supposed to be paying attention to your social life. This is not how it was described in any immunology textbook written before the late 1990s. The immune system had a job: pathogens, foreign bodies, cellular damage, the surveillance and elimination of things that didn't belong inside the organism. It was a biological defense network, intricate and ancient, but bounded. It dealt with the physical interior. What happened in the social world was somebody else's problem. And then the data started arriving, and the boundary turned out to be less solid than assumed. Interleukin-6 is a cytokine, a small signaling protein released primarily by immune cells, though also by other cell types including neurons and fat cells. It coordinates the inflammatory response. When tissue is damaged or infected, IL-6 rises, recruiting immune cells to the site, triggering the liver to produce acute phase proteins, raising body temperature, accelerating the response. It is a fast, effective, well-calibrated molecule for the job it was built to do. In lonely individuals, IL-6 is elevated, not dramatically, not at the levels seen in active infection or acute injury, but measurably, consistently above the levels seen in non-lonely individuals matched for age, health status, and objective social contact. That last part matters. The elevation appears independent of how many people the person actually interacts with. It tracks the subjective sense of isolation, the perception of disconnection, the feeling of being insufficiently connected, regardless of whether, by any external measure, the person is actually alone. The immune system is responding to a feeling. Steve Cole at UCLA has spent years mapping what he calls the social regulation of gene expression, the ways in which social experience alters which genes are switched on and off in immune cells. His work identified a pattern he termed CTRA, the conserved transcriptional response to adversity. In individuals experiencing chronic threat, including social threat, including loneliness, a specific cluster of gene expression changes occurs in immune cells. Genes involved in inflammatory response are upregulated. Genes involved in antiviral defense, the type 1 interferon response, are downregulated. This is the same pattern seen in animals exposed to physical danger. The body, preparing for the possibility of injury in a hostile environment, shifts its immune resources toward wound response and away from infection resistance. Inflammation up. Antiviral capacity down. It makes evolutionary sense. If you're about to be attacked, wounds are the likely threat. Bacteria entering through her broken skin, bleeding, tissue damage. The inflammatory response handles these. Viruses, transmitted through close social contact, through breath and proximity, are more relevant when you are embedded in a group, when other bodies are near. Alone, the logic goes, you are less likely to encounter a virus and more likely to encounter a predator. The immune system read the social signal and rearranged its priorities accordingly. What it cannot know, what no part of this ancient system can know, is that the loneliness driving that rearrangement is happening in a third-floor apartment with a locked door and central heating and no predators within several thousand miles. The CTRA pattern doesn't ask for context. It reads the social signal, applies the response and proceeds. C-reactive protein is another marker. CRP is produced by the liver in response to IL-6 signaling and is one of the standard measures of systemic inflammation used in clinical medicine. Elevated CRP is associated with cardiovascular disease risk, with metabolic syndrome, with a range of conditions involving chronic low-grade inflammatory burden. In population-level studies, loneliness is associated with elevated CRP, again, tracking the subjective experience rather than objective isolation. The relationship is not enormous in effect size, but it is there, across multiple studies, in multiple populations, and it accumulates. Low-grade chronic inflammation is not an acute event. It doesn't announce itself. It operates over years, gradually altering the biochemical environment of the cardiovascular system, the brain, the metabolic organs. The elevated IL-6 that a lonely person carries today is not causing a heart attack tomorrow. but the same elevation, sustained across months and years, contributes, quietly, incrementally, to a biological environment where certain disease processes find it easier to take hold. There is something I keep returning to in Cole's work, and I'm genuinely not sure what to do with it. The CTRA pattern, so the shift toward inflammation away from antiviral defense, was also found to be partially reversible. Social reconnection in some studies shifted gene expression back toward the baseline pattern, not instantly, not completely, but measurably. The immune system had changed in response to perceived isolation, and it changed again in response to perceived connection. Which means the immune cells circulating in your blood right now are, in some molecular sense, carrying information about your social experience. Not a record of specific relationships or specific losses, but a set of transcriptional states, genes on, genes off, that were set in part by whether you felt sufficiently connected to other people over recent weeks and months. The body keeps accounts in places you would not think to look. Something about the arms, the skin there, the way it carries temperature differently than the face, slower to warm, slower to cool. A bodily fact that has no particular relevance here, and yet arrives anyway. NF-kappa B is the transcription factor primarily responsible for switching on the pro-inflammatory genes in Cole's CTRA pattern. It sits inside immune cells, activated by signals from the cell's surface receptors, and when active it moves into the nucleus and begins transcribing inflammatory proteins. It is a downstream effector of the stress response, norepinephrine, released by the sympathetic nervous system under threat conditions, can activate NF-kappa B signaling in immune cells. So the pathway from perceived social threat to inflammatory gene expression runs roughly like this. Social isolation is perceived, the brain reads it as threat, the sympathetic nervous system activates, norepinephrine reaches immune cells, NF-kappa B is activated, inflammatory genes are transcribed. A feeling, the felt sense of not being adequately connected, becomes a change in which proteins immune cells are making. The social world is being translated continuously into molecular biology. What I find hardest to fully absorb about this, and I've read Cole's papers multiple times, is not the mechanism. The mechanism is, in its own way, logical. It is the specificity of the trigger, not objective aloneness, not measurable social contact, the subjective sense of isolation. Two people with identical social networks, identical frequency of contact can have different inflammatory profiles based on how connected they feel. The immune system is not counting your friends. It is listening to something more interior than that, something closer to the felt quality of your relationships, whether they register at some level below language as genuinely present or quietly absent, and it responds accordingly, with molecules, with gene expression patterns that have been conserved across vertebrate evolution for longer than the human species has existed. Listening in its ancient and completely unsentimental way to something the rest of medicine is only recently beginning to take seriously. You're still right now, not inactive. The heart is moving, the lungs are moving, the gut is doing its slow work, but the body's relationship to the external world has narrowed. The room is quiet, the body has stopped performing. This is one kind of stillness. The autonomic nervous system is running a continuous assessment of the environment. Not consciously, not voluntarily, it reads light levels, sound, the presence or absence of other bodies, the quality of the space, and based on what it reads, it selects a state. There are broadly 3 states available. The first is the one associated with safety and social engagement, sometimes called the ventral vagal state. governed by the myelinated vagus nerve and connected to the muscles of the face, throat, and middle ear. Heart rate is flexible here, sirs. Breathing is easy. The face is capable of fine expression. The body is, in a specific physiological sense, open. The second is sympathetic activation, threat-oriented, mobilizing the system that raises cortisol and readies the muscles. The third is a deeper withdrawal, a shutdown quality, the dorsal vagal state, associated with immobilization, with the particular flatness of prolonged overwhelm. Most people move between these states many times a day without noticing. What determines the transition is not always obvious. A tone of voice, a shift in ambient sound, the presence or absence of another person's breathing nearby. Right now, your body is in one of these states. The one it has chosen depends on what the nervous system has concluded about whether this moment is safe, and that conclusion is not purely rational. It is based on accumulated signal, relational history, recent experience, the particular quality of tonight's quiet. There is a difference between silence that follows connection and silence that precedes it, not poetically, physiologically. Stephen Porges, who developed polyvagal theory, documented the ways in which the middle ear musculature, governed by some of the same cranial nerves involved in social engagement, tightens under threat conditions and relaxes under safety. When the ventral vagal system is active, the middle ear preferentially filters for the frequency range of human speech. The nervous system is literally tuning itself to receive other people. When that system is not active, when the environment has been read as insufficiently safe, insufficiently connected, the middle ear filtering shifts. Background sounds become more prominent. The specific frequency range of human voice recedes. The quiet sounds different, depending on which state the nervous system is in. You may have noticed this. The silence of a room where someone you trust has recently been feels different from the silence of a room that has been empty for a long time. Not psychologically different, differently textured. The body is doing something different in each. The weight of the blanket, if there is one, the specific pressure of it across the chest and feet, something real and present and entirely unrelated to all of this. The autonomic nervous system doesn't categorize silence as safe or unsafe based on the silence itself. It categorizes it based on what the relational context has been. A person who has spent recent weeks feeling genuinely connected to others will, on average, bring a different autonomic baseline to a quiet room than a person whose relational environment has felt thin or unreliable. The silence lands in a differently prepared nervous system. One reads, rest. The other reads, alone again. Neither reading is chosen. Heart rate variability is measurably different between these states. The variability, the flexibility with which the heart responds to moment-to-moment demands, is higher when the ventral vagal system is active. The heart is more responsive, more capable of range, in states of chronic threat perception, including the low-level threat of loneliness. Vagal tone is reduced. Heart rate variability compresses. The system is holding a position rather than moving freely through its range. This is measurable with the sufficiently sensitive device. It is also, if you know what to look for, faintly perceptible from the inside. The stillness of the body right now is either resting or bracing. Probably something between those. Most moments are. The nervous system is still checking, even now, still reading the ambient information for social signal. Still asking, in its wordless way, whether the quiet around you is the quiet of safety or the quiet of absence. It asks this continuously. It does not stop asking when you fall asleep. What it concludes shapes the biology of the next hours, the cortisol curve, the depth of slow-wave sleep, the particular quality of rest that may or may not arrive by morning. Not catastrophically, Not irrevocably, just differently. Depending on what the stillness in this particular body on this particular night sounds like. When the brain is not doing anything in particular, it does not go quiet. This is one of the stranger discoveries of modern neuroimaging, that the resting brain, the brain between tasks, the brain that appears to be doing nothing, is in many ways more active than the brain solving a problem. A specific network of regions lights up reliably whenever attention is not directed outward. Researchers initially called it the default mode network because it seemed to be the brain's default state, what it returns to when released from demand. The medial prefrontal cortex, the posterior cingulate cortex, the angular gyrus, the hippocampus. These regions, distributed across the brain but functionally tightly coupled, activate together when you are not doing anything, when you are lying still, letting thoughts arrive without direction, when you are, as you might be right somewhere between wakefulness and drift. What the DMN is doing during this apparent rest turns out to be largely social. It is simulating other people, not always deliberately, not always consciously, but the default activity of the resting human brain is, in significant part, the mental modelling of other minds, imagining what someone else is thinking, replaying a conversation and reconsidering what it meant, constructing a possible version of a person you're going to see tomorrow, and running through how that might go. The brain, left to its own devices, goes immediately and persistently to other people. This has an evolutionary logic that feels almost too obvious to state. Social species need to predict the behavior of other members. The brain that uses its downtime to run social simulations, to update its models of who is safe, who is unreliable, what the group dynamics currently look like, has a survival advantage over the brain that simply idles. The DMN is not resting. It is rehearsing. There is something I find quietly remarkable about this. Not surprising exactly. Once you hear it, it feels inevitable. But remarkable in the way that confirms something you half knew. That the mind, when it wanders toward people. That the texture of an idle afternoon is largely made of other people, imagined and remembered and anticipated. That even in the apparent privacy of your own drifting thoughts, you are probably at the cellular level running social simulations. You're probably doing it right now, actually. Some version of it, some face or voice or anticipated interaction threading through the edge of whatever this is becoming. In lonely individuals, the default mode network functions differently at rest. The finding is not that it becomes less active. The regions still light up, the network still runs. What changes is the nature of the activity. Resting state functional connectivity analyses, measurements of how tightly coupled the DMN regions are during rest, show alterations in lonely individuals that suggest the network is operating within a narrower range. The social simulation is still happening, but its quality shifts. Danilo Bizdock at McGill University led some of the largest structural and functional studies of loneliness in the human brain, studies involving sample sizes roughly 100 times larger than earlier work, drawn from the UK Biobank. What emerged, across multiple analyses, was a picture of the DMN in loneliness as operating with reduced flexibility. The connections between regions were present but running differently. The posterior cingulate, involved in self-referential thought and the integration of past experience into current judgment, showed altered connectivity patterns. The medial prefrontal cortex, which is centrally involved in thinking about other people's mental states, their beliefs, intentions, the particular texture of their inner life, was implicated throughout. The specific thing that seems to contract in the lonely DMN is not social thought itself, but the range of social possibility it generates. Non-lonely individuals, running their default mode social simulations, produce a wider variety of social scenarios. Other people arrive in these simulations with more complexity, more ambiguity, more potential. The mental models are less fixed. In loneliness, the simulations narrow. Other people become, in the default mental model, somewhat less available, somewhat less reliably positive. The range of imagined social futures contracts toward the more cautious end. The brain, running its rehearsal, is rehearsing a social world with a particular cast, one where others are present but somewhat withheld, not hostile, not absent, just not quite fully there. This connects to the 120 millisecond threat detection from earlier in a way that feels important, though the research hasn't fully drawn the line between them. The fast threat detection happens in real-time social encounters. The DMN shift happens in the spaces between encounters. Together they suggest something like a continuous recalibration, the nervous system updating its social world model, both in the moment of contact and during the quiet hours of resting thought. Both processes pulling gently and persistently in the same direction. The specific texture of lying still and not quite sleeping, thoughts arriving half-formed, images with no narrative, the mind visiting faces without choosing to, is largely the DMN running. The hippocampus stitching together fragments of memory and experience. The posterior cingulate evaluating their relevance to the current sense of self. The medial prefrontal cortex doing its quiet work of modelling other people. What it models, and how generously, depends on what it has learned to expect. A person who has felt over recent months that other people are mostly available, mostly warm, mostly responsive, brings a particular default model to that drifting state. The simulations run with a certain ease. Social imagination has room to move. A person whose relational experience has been thinner, or more uncertain, or characterized by the particular quality of feeling present in rooms without being reached. That person's DMN is running the same process on different data. The simulation engine is intact. What feeds it has changed. I tend to think this is one of the more underappreciated aspects of what chronic loneliness does. Not the acute pain of it, the empty evening, the unanswered message, but the slow recalibration of what the resting mind imagines is possible. The way the social world in the DMN gradually becomes a slightly smaller place, not through any decision, not through pessimism or distortion exactly, just through the honest updating of a system that learns from experience. The brain is doing its job. It is modelling reality as accurately as it can, given the data it has been given. The problem is that the data, sustained over time, changes the range of what the model considers likely. And the model, once set, shapes what you notice, what you expect, what you allow yourself to want from the next social encounter before it has even begun. The DMN is still running now. Whatever drifting is happening at the edge of your attention, whoever arrived there unbidden, whatever version of a conversation you've been quietly revisiting, that is the default mode doing what it was built for. Modelling, rehearsing, keeping its slow cellular account of the social world as it has come to understand it. Whether that account is generous or cautious, open or contracted, is something the biology carries forward into every room you walk into, every silence you try to read, every stranger whose face, in under 150 milliseconds, the nervous system has already begun to assess. The skin is the body's largest organ, which is one of those facts that gets repeated so often it stops landing. But it is worth recovering what it actually means, that the boundary between you and the world is an enormous, living, constantly sensing surface. Not passive, not simply a container, but an active input system feeding the brain a continuous stream of information about what is making contact with it and how. Most of this information is processed through fast myelinated nerve fibers. Sharp, precise, immediate, the type that tells you exactly where your finger is, what texture it's touching, whether something is too hot. This is the ordinary sensory system, efficient, accurate, primarily informational. But there is another system running alongside it. Slower, less precise, concerned not with the location or texture of touch, but with something harder to name, the quality of it. Whether it is the kind of touch that means something. See, tactile afferents are unmyelinated nerve fibers found specifically in hairy skin. The arms, the back, the sides of the neck and face, they are absent from the palms of the hands and the soles of the feet. The surfaces most involved in manipulating and navigating the physical world. Their distribution is not random. It maps roughly onto the skin surfaces most likely to receive contact from another person. They respond to a specific velocity of touch, roughly 1 to 10 centimeters per second, light pressure moving slowly, the speed of a hand drawn gently across a forearm, not a firm grip, not a fast brush, the particular pace of deliberate, unhurried contact between people who are not being careful around each other. Outside that velocity range, their response drops sharply. Too fast and they barely fire. Too slow and the signal fades. They're exquisitely tuned to one thing, and that thing is the specific signature of effective touch. Touch that carries intention. When CT afferents fire, they do not send signals along the usual somatosensory pathways to the primary sensory cortex. They project instead to the posterior insula, the same region involved in interoception, in the monitoring of the body's internal state, in the felt sense of being a body that is currently okay. From there, the signal reaches limbic structures. Reward circuits, the systems involved in emotional regulation and social bonding. The skin, in this specific situation, is not sending the brain a tactile report. It is sending a social one. Francis McGlone at Liverpool, John Moores University has spent decades working on the CT afferent system, and one of the consistent findings from his group is that the signal from these fibers produces activation in the insula that correlates with pleasantness ratings, and that the pleasantness is specifically social in character. The same touch applied by a stranger and by a trusted person produces different cortical responses. The CT system is sensitive not just to velocity, but through some combination of contextual processing, to who is doing the touching. The nervous system has built into the skin itself a detector for a particular kind of connection. There is something almost too specific about this. The velocity window, the distribution on hairy rather than glabrous skin, the projection to the insula rather than the sensory cortex. Every detail of the CT afferent system looks, in retrospect, like a design for receiving exactly one thing, the slow, deliberate touch of another person who means it. A system this specific, this anatomically committed, implies that what it detects has been important for a very long time. The developmental literature bears this out in ways that are, depending on how you hold them, either clarifying or difficult. Harry Harlow's mid-20th century work with rhesus monkeys, infant monkeys given the choice between a wire surrogate that provided food and a cloth surrogate that provided only soft contact, established something that seems obvious in retrospect but required the experiment to make concrete. Contact comfort is not a secondary consequence of feeding. It is a primary need. The infants chose the cloth mother. They ran to it when frightened. They used it as a secure base. In human infants, tactile stimulation, being held, stroked, carried, is associated with the regulation of cortisol, the development of the vagal system, the calibration of the stress response. Premature infants who receive kangaroo care, sustained skin-to-skin contact with a caregiver, show better autonomic regulation, faster weight gain, more developed sleep architecture than those who do not. The nervous system is not simply comforted by touch. It is, in early life, partly organized by it, which raises a question that the research has not fully answered. What happens to a nervous system that is organized in the presence of effective touch and then, at some later point, receives substantially less of it? Not none necessarily, just less than the system was calibrated to expect. The short answer, from what the literature suggests, is that the CT afferent pathway remains present and functional throughout life. The fibers don't disappear. The insula doesn't lose its capacity to receive the signal. But the effect of prolonged reduced input appears to be something like a shift in the system's sensitivity. The nervous system's baseline state adjusting to accommodate what it is actually receiving rather than what it was built to process. This is speculative at the edges. The research on long-term touch deprivation in healthy adults is not vast. But what exists suggests that the effective touch system does not simply idle patiently in the absence of input. It recalibrates. The threshold for perceiving touch as social and meaningful may shift. The insula's response may dampen. The specific reward quality of slow, deliberate contact, the thing CT afferents evolved to report, may arrive less fully when the pathway has been underused. The weight of a blanket is not the same as the weight of a hand. The body knows this in the specific way that nervous systems know things, not consciously, not in language, but in the different quality of response each produces. The blanket applies pressure to the skin and the mechanoreceptors report it faithfully. But the CT afferents are quiet. They are waiting for a velocity, a direction, a contextual signal that tells them the contact is coming from someone. Right now, if you are lying still, with your skin's in contact with fabric, with air, with your own body where limbs rest against each other, all of this is registering. None of it is reaching the insula through the effective touch pathway. I want to be careful here not to overstate this. Touch deprivation is not the same as loneliness, and the absence of effective touch is not the only thing loneliness involves. The opioid system, the HPA axis, the insular cortex, the DMN, all of these are running their own parallel accounts. Touch is one channel among many, but it is a channel with its own dedicated hardware, built into the skin, projecting to exactly the structures that regulate emotional state, stress response, and the sense of bodily safety. And when that channel is consistently underactivated, the nervous system is running without an input it specifically evolved to receive. Not broken, not dysfunctional, just missing a signal that at some level of its architecture it is still waiting for. What touch leaves behind when it has been present is not a memory exactly, it is a calibration, a set point. The nervous system having learned, through the CT afferent pathway and everything downstream from it, what a certain quality of contact feels like and what it produces in the body. The insula having received that signal enough times to know what it means. And in its absence, that calibration persists. The system doesn't forget what the signal was, it simply waits, in its patient biological way, for the input to resume, the skin right now registering what is there and what is not. There is a quality that belongs to a body that has been still for a long time, not the stillness of just lying down, that has its own particular restlessness, a residue of the day's activities still moving through the muscles. This is something further in, when the body has been horizontal long enough that it has stopped anticipating movement, when the weight of it has settled fully into whatever surface it's resting on, and the joints have released their held positions, and even the small habitual tensions of the face have softened. You may be approaching that now. This chapter doesn't introduce anything new. What it does is stay with what has already been covered, not to summarize, not to conclude, but because the biology of loneliness is not a list of separate findings. It is a single condition distributed across many systems simultaneously, and sometimes the most honest thing is to simply let that distribution be felt rather than explained. The anterior cingulate is still there, behind the forehead, in whatever state it's in tonight. The opioid receptors are running their quiet account. The HPA axis has set its cortisol slope for the hours ahead. Whether that slope will be steep enough to allow full descent into slow-wave sleep, or whether it will stay elevated, pulling the body back towards surface before it reaches depth, that has already been determined, largely, by the week and the month and the particular relational texture of recent experience. None of this requires attention to proceed. It's already in motion. If you've found something in these episodes that feels worth returning to, that's what the subscribe button is for, genuinely. Not a transaction, just a way of finding this place again when you want it. What strikes me, having spent time with this biology from many directions, is how thoroughly distributed the condition is. There is no single loneliness region, no one structure that, if changed, would resolve the whole thing. The anterior cingulate and the insular and the dorsal Raphe nucleus and the ventral striatum and the prefrontal cortex and the immune cells expressing their inflammatory genes and the CT afferents waiting in the skin. All of them are running, all of them are adjusted, all of them are doing what the organism's biology determined was appropriate given the social conditions it has perceived. They are not malfunctioning. That is the part that keeps returning. They are functioning exactly as designed, in a world that the design did not anticipate. The building metaphor arrives here and only once. It is not that any single room is dark, it is that the whole building is lit differently. The same rooms, the same wiring, the same switches, but the quality of light throughout has shifted in a way that is difficult to identify from inside a single room and only becomes visible when you step back and look at the whole structure at once. That is what the neuroscience of loneliness looks like from a sufficient distance. Not damage, not darkness, just different light. A different setting on every system, each one reasonable in isolation. The combination of them producing something that, sustained long enough, costs the organism more than it can easily afford. The dorsal Raphe is keeping its account. The substantia nigra has its readiness. Somewhere in the immune cells, NF-kappa B is in whatever transcriptional state the social signal has placed it. The default mode network is running its simulations, narrower or wider depending on what the accumulated data of recent experience has taught it to expect. All of this right now in the body that is lying still. If any of this has felt worth something, if one episode settled something in you or named something that didn't have a name before, that's what the membership is for. A small quiet way of keeping this space going. And if a super thanks has ever crossed your mind, that genuinely matters more than it probably sounds. The skin is in contact with fabric. The CT afferents are quiet. The middle ear is in whatever filtering state the autonomic nervous system has selected for tonight's particular quality of silence. The insula is receiving its body report, temperature, weight, the slow rhythm of breathing, and integrating it into the felt sense of being here, in this body, in this room, at this hour. Whether that report reads as safe or as merely still depends on things the biology has already decided. There is no resolution available in a single night's biology. The systems that have been described across these chapters did not arrive at their current states quickly, and they will not leave them quickly either. The CTRA pattern in the immune cells takes weeks to shift. The white matter pathways take longer. The DMN social simulation range recalibrates gradually across many experiences, many encounters that go differently than the model predicted. What does shift quickly, what the nervous system is actually capable of updating in something close to real time, is the autonomic state. The ventral vagal or sympathetic or dorsal vagal setting, the cortisol slope for the next few hours, the quality of the sleep that is coming. These are not nothing. They are the immediate layer. And the immediate layer is the one you are actually in right now. The body that has been described across 15 chapters of biology is your body. Not as metaphor, not as a general human body standing in for yours, the anterior cingulate that registers social pain as physical pain. That is yours. The dorsal raphe that tracked your isolation and built its readiness for reunion. That is yours. The CT afferents in the skin of your arms, waiting at their particular velocity threshold. Those are yours. All of it running right now at whatever settings the accumulated experience of your life has placed them on. And all of it. Every system, every pathway, every molecular state capable of being shifted. Slowly, by the quality of contact over time. If you press like on this, and I mean this without any pressure, it tends to bring it to someone else who might need to lie still with this biology for a night. That's the only reason it matters. The body has stopped anticipating movement. The room is what it is. The silence has whatever frequency it has tonight. Safe or watchful, or somewhere in the large space between those states. The biology will run its night cycle regardless. The cortisol will do what it does. The glymphatic system will attempt its clearance. The DMN will send its drifting simulations through whatever range it has learned to occupy. None of this requires anything from you. The anterior cingulate is quiet now, or quieter. The opioid system is in its state. The HPA axis is settling or not settling, depending on what the nervous system has concluded about whether tonight is safe. Somewhere in the brainstem, the dorsal raphe is simply there, ancient, small, doing its particular job in the dark, and the skin, the enormous, patient, carefully instrumented surface of it, is resting against fabric, against air, against the ordinary contact of a body that is simply here, waiting, as all living tissue waits, for whatever the next few hours in their quiet biological way decide to bring.